Okay, let's get started. It's really quite a statistic when you think about it.
Mm-hmm.
Nearly half of drugs pulled from the market. It's because of heart problems. Things like cardiotoxicity, arrhythmias.
It really is. And that's even with all the advances we've made, like in nanomedicine.
Exactly. It just shows how tricky it is to predict how a new therapy will actually interact with the human heart. It's complex.
Absolutely. And these withdrawals, they're not just scientific setbacks. They cost a fortune. And maybe more importantly, they delay potentially vital treatments getting to patients.
Right. Which brings us to, well, a pretty unexpected place for today's deep dive. We're not starting with complex human trials today.
No.
No, we're going much, much smaller. Down to a tiny worm, Caenorhabditis elegans.
Well,
It's only about 1 millimeter long.
C. elegans, the nematode.
That's the one. And I know what you might be thinking listening to this. A worm.
Yeah.
Seriously. What can that tell us about the human heart?
It does sound a bit counterintuitive at first glance.
It really does. But that's the aha moment we're after today. We're exploring how scientists are actually using this seemingly simple creature to get vital clues about how new drugs, new therapies might affect our hearts.
And if you think about the bigger picture, the drug development pipeline, you can see the appeal immediately.
How so?
Well, the traditional route is just so long, incredibly expensive, and let's be honest, has a really high failure rate.
Yeah.
So finding reliable ways to spot potential problems early on, that's hugely valuable. It could streamline the whole process.
Makes sense. And guiding us through this microscopic world is a study from ACS Nano. It's titled Arrhythmic Effects Evaluated on Caenorhabditis elegans, the case of polypyrrole nanoparticles.
Okay.
And what's really neat is they used C. elegans not just for these experimental nanoparticles, polypyrrole nanoparticles, or Ppy NPs, but also to check its response against well-known heart medications.
So a validation step. That's smart. It addresses that initial question directly, right? Why this worm?
Exactly. What makes C. elegans a good model for studying these kinds of heart-related effects?
That's the million-dollar question.
So that's our mission today.
Yeah.
Figure out why this tiny worm is useful for predicting potential heart side effects, and specifically, what this study found out about those polypyrrole nanoparticles. Ready to dive in?
Let's do it. So why C. elegans?
Yeah, why the worm?
Well, like we touched on, the traditional drug development path has major limitations. Cost, time, failure rates.
Right.
Model organisms offer a potential bridge. They sit between, say, cell cultures in a lab dish and full human trials.
Kind of like a stepping stone.
Exactly. And it fits with the whole 3Rs principle in animal research, too. Replace, reduce, refine, using simpler organisms where possible.
Okay, so using worms might reduce the need for, say, testing on larger animals later down the line. But there are other small models, right, like fruit flies, zebrafish. Why C. elegans specifically?
Good question. Zebrafish and fruit flies are definitely valuable tools, no doubt. But C. elegans has some pretty unique advantages. Okay, well, first off, and this is kind of mind-blowing, genetically, they share a surprising amount with humans.
Really? A worm?
Yeah. Many fundamental biological pathways are conserved. So, what happens in the worm can actually give us clues about human biology. Plus, they have a super short lifespan, reproduce incredibly quickly.
So you can study effects across generations fast and get large numbers.
Precisely. Large sample sizes, quick turnaround.
Yeah.
And you can actually freeze down different mutant strains and store them for ages. That's invaluable for studying specific genes. And maybe one of the most striking things, they're transparent. Completely see-through, even as adults.
So you can just look inside, see what's going on.
Pretty much. You can observe internal structures, biological processes, how drugs are distributed.
Yeah.
It's a huge advantage for observation.
Okay, the transparency thing is cool. You mentioned a key similarity to the heart earlier, even though it doesn't really have a heart like ours.
Right. No heart, no circulatory system. But it has a pharynx. It's this muscular tube it uses for feeding, kind of like its throat pump. And the muscle cells in this pharynx, they share some really fundamental features with human heart muscle cells, cardiomyocytes.
Like what?
Well, both are muscle cells, obviously, and they're connected to nerve cells through things called gap junctions. These are like tiny communication channels between cells.
So similar plumbing in a way. What about the electrical side, the signals?
Exactly, that's key. Both the worm pharynx cells and human heart cells use similar voltage-gated ion channels. These are pores in the cell membrane that control the flow of charged ions, sodium, potassium, calcium.
And that flow creates electrical signals.
Yes, action potentials, which then trigger the muscle to contract. The basic mechanism is conserved,
Huh?
and it goes deeper. C. elegans has genes that are orthologous to human heart genes,
Orthologous meaning?
meaning they evolved from a common ancestral gene and a shared ancestor way back when, and often they still have similar functions.
Got it. Any specific examples relevant here?
Yes, the paper highlights a couple. The worm gene UNC68 is related to human genes for ryanodine receptors, which are crucial for calcium release in heart muscle. And the CSQ1 gene relates to human calsequestrin genes, also involved in calcium handling.
And why pick those two? Are they linked to heart conditions?
Absolutely. In humans, mutations in those specific genes, the ryanodine receptor and calsequestrin genes, can cause a serious arrhythmia called CPVT, catecholaminergic polymorphic ventricular tachycardia.
Okay.
And here's the kicker. The researchers point out that worms with mutations in their corresponding genes, UNC68 and CSQ1, show effects in their pharynx that look remarkably similar to CPVT.
Wow. Okay. That is a strong parallel. So researchers can measure things in the worm's pharynx that might predict human heart effects.
That's the idea. They can easily measure the pharyngeal pumping rate, how fast it's beating, and they can also measure calcium levels inside those pharyngeal cells.
And changes in those could indicate a drug's potential effect.
Potentially, yes. The pumping rate is a fairly direct measure of muscle activity, and calcium signaling is fundamental to contraction in both systems.
Okay, this makes the worm seem much less random now.
Yeah.
So the first step for these researchers was proving the model works, right?
Right.
The validation you mentioned.
Exactly. Before testing the new nanoparticles, they needed to show that C. elegans responds appropriately to drugs with known human heart effects.
What did they use?
They picked two well-established FDA-approved drugs. Propranolol, this study calls it PL.
The beta blocker, right? Slows heart rate.
Yep. And racepinephrin, or RE, which is a beta-adrenergic agonist, does the opposite, speeds up the heart rate.
Okay, so two drugs with clear opposing effects on the heart. How do they test them in the worms?
They exposed worms at a specific developmental stage L4 larva to a set concentration, 100 micromolar, of either PL or RE for 24 hours. Then they looked closely at the outcomes.
Did the worm pharynx behave like a human heart would?
It really did. First, importantly, neither drug seemed toxic in a general sense. Worm survival, growth, development, even reproduction all looked fine.
Okay, so they tolerated the drugs.
Yes. But then, looking at the pharyngeal pumping rate, propranolol decreased the rate.
That's like it slows human heart rate.
Exactly. And racepinephrin increased the pumping rate.
Matching its effect in humans. That's actually pretty neat.
It's quite convincing, isn't it? It strongly suggested that, yes, this worm model is relevant for these kinds of cardiac-like effects.
Did they notice anything else? Like how long the effects lasted?
They did. They found the effects were time-dependent. The biggest change in pumping rate was seen after the full 24 hours of exposure, and also the effects were reversible. When they took the drugs away, the pumping rates gradually went back towards normal. Though, interestingly, the effect from racepinephrin wore off quicker than propranolol's effect.
Okay, so validation successful. The model seems to work. Now, onto the main event. These polypyrrole nanoparticles, the Ppy NPs. What's the interest in these?
Well, Ppy NPs are part of this emerging field of conductive nanomaterials for biomedical use. And specifically for the heart, some lab studies in vitro work suggested they might be good for arrhythmias.
So,
Those studies indicated they could potentially increase the speed of electrical signals in heart tissue, maybe help synchronize irregular rhythms, and they also seem to enhance calcium signaling in heart cells.
sounds promising. But you mentioned earlier there were some disagreements about safety.
That's right. It's a crucial point. While some studies suggested they were biocompatible, safe for living systems, others raised concerns, especially at higher doses.
Like what kind of concerns?
For instance, some previous work using zebrafish showed high levels of certain Ppy NPs could cause developmental problems and even slow the heart rate. So definitely conflicting data out there.
Which makes a thorough assessment in a living organism in vivo really necessary.
Exactly. You need to understand how they behave in a whole biological system. Hence, turning to C. elegans.
So how did they make and check these specific Ppy NPs for the worm study?
They used a standard chemical synthesis method. The resulting nanoparticles were mostly spherical, around 130 to 200 nanometers in size.
Tiny.
Very tiny. And they did extensive checks: size, shape, chemistry, and importantly, stability. not just in water, but also in solutions designed to mimic the conditions inside the worm's gut, like different pH levels.
And they hold up okay?
Generally, yes.
Yeah.
They seemed stable enough for the experiments.
So they exposed the worms. How did they know the worms actually ate the nanoparticles?
Simple observation. Because the worms are transparent, they could just look under a microscope after 24 hours and see the dark nanoparticles accumulated inside the worm's intestines.
Ah, the transparency advantage again.
Right.
Seeing is believing.
Pretty much.
Did they figure out what happened to the MPs and said did they change it all?
Yes, they looked into that too. They actually developed a method using bleach to carefully dissolve the worms and recover the nanoparticles that had been inside.
Whoa.
Yeah. And then they analyzed those recovered MPs using powerful microscopy, TEM, and light scattering, DLS. They found evidence that some of the nanoparticles had started to dissolve slightly and also clump together aggregate within the gut environment.
Interesting. So they weren't just passing through unchanged. They were interacting.
Seems like it, yes.
Okay. So the worms ate them. They changed a bit inside. What about the overall impact on the worm? The systemic effects? Toxicity?
That was the next big check. They looked at various toxicity markers after 24 hours of exposure to different Ppy NP concentrations.
What were the key findings there?
Well, really encouragingly, they saw no negative impact on worm survival, even at the highest dose they tested, 500 micrograms per milliliter.
That's good.
And as the main concentration they used for other tests, 100 micrograms per milliliter, there was also no significant effect on growth or development.
What about reproduction? Or effects on the next generation?
Nope, nothing significant there either. The number of offspring was normal, and the next generation, the F1 worms, also grew normally. So no obvious reproductive or intergenerational toxicity.
Okay. Sounds pretty biocompatible so far in the worms.
Yeah.
Did they look for more subtle things like stress or changes in metabolism? Fat storage?
They did. They used special stains and even a sophisticated technique using synchrotron light SR-FTIR to look at the worm's chemical makeup.
Fancy. What did that show?
No signs of major lipid accumulation, unusual fat storage, or oxidative stress, which is like cellular damage. But they did see hints of increased lipid metabolism, like the worms might be burning through fats a bit faster.
Why would that be?
Their hypothesis was it might be linked to that slight increase in pharyngeal pumping they saw. More pumping means more energy needed, potentially leading to increased fat metabolism.
Ah, okay. A possible functional consequence, not necessarily toxicity.
Exactly.
So let's focus on that pharyngeal effect then.
Yeah.
You said there was a slight increase in pumping in normal wild-type worms.
Yes, a small but measurable increase at that 100 microgram per milliliter concentration after 24 hours.
But the really interesting stuff happened with the mutant worms.
That's where it got quite striking, yes. They tested 2 mutant strains known to have slower than normal pumping rates.
Remind me which ones.
One was JD21, which has a mutation in that HEK1 gene, the one related to human T-type calcium channels.
Make the calcium connection.
And the other was DA464 with a mutation in EAT5 related to those gap junction communication channels.
Okay. And how did they respond to the Ppy NPs?
Both showed an increase in their pumping rate. But the JD21 strain, the calcium channel mutant, showed a much more significant increase. It was the most dramatic effect they saw.
Wow. So the NPs had a bigger effect on the worms already deficient in calcium channel function.
It certainly looks that way. The DA464 gap junction mutant also sped up, but not nearly as much as JD21.
What does that difference suggest?
Well, the fact that the biggest effect was in the calcium channel mutant strongly points towards calcium signaling being involved in how these Ppy NPs work. It suggests the NPs might be somehow compensating for or interacting with that defective calcium transport.
Did they look at the timing again? Was it immediate or did it build up? And did it last?
Yes, they tracked it over time. In all 3 strains, wild type and the 2 mutants, the increase in pumping rate generally occurred progressively during the 24-hour exposure.
Okay.
And interestingly, the increased rate persisted for quite a while even after they removed the nanoparticles. It took up to 72 hours, 3 days, for the pumping rates to return completely to normal.
So a relatively sustained effect, not just transient.
Exactly.
Now, you said the big effect in the JD21 strain pointed towards calcium. Did they investigate that directly? Look at calcium levels.
They did. That was the next logical step. They used worms that were genetically engineered to have a fluorescent calcium sensor, specifically in their pharynx muscles.
So they could literally watch the calcium ebb and flow.
Essentially, yes. It allows real-time monitoring of calcium ion fluctuations, these things called calcium transients during the pumping cycle.
Very cool tech. What did the calcium sensors reveal?
Okay, so In the normal, wild-type worms, adding the Ppy NPs did two things to the calcium signals. First, it increased the frequency of the calcium spikes.
More spikes per minute, matching the faster pumping. Makes sense.
Right. But second, it also increased the width or duration of each calcium spike, meaning calcium levels stayed higher for longer during each contraction cycle.
Okay. Longer calcium elevation. What about in the mutant worm, JD21, the one with the calcium channel issue?
This is the crucial part. In the JD21 mutants, the Ppy NPs still increase the frequency of calcium spikes, consistent with the faster pumping rate they observed.
Okay.
But the nanoparticles did not affect the width or duration of the calcium peaks in these mutants. That remained unchanged.
So the MPs could make the mutant pump faster, but couldn't prolong the calcium signal like they did in the normal worms.
Precisely.
What does that tell us? That seems like a really specific finding.
It strongly reinforces the idea that the T-type calcium channel, the one affected in the JD21 mutant, coded by a G01, is playing a critical role in how these Ppy NPs influence pharyngeal activity. The effect on spike duration seems dependent on that specific channel.
So the mechanism seems tightly linked to modulating calcium signaling, potentially through interaction with these T-type channels.
That's the main conclusion they drew from the calcium imaging, yes.
Okay, let's try and pull this all together then. What are the big takeaways from this deep dive into worms, nanoparticles, and heart effects?
Well, takeaway number one is that C. elegans, this tiny worm, really showed its potential as a valuable, relatively simple model for doing initial screens for potential heart-related effects.
The validation with propranolol and racebinephrin really hammered that home.
Exactly. It accurately reflected their known effects in humans, which builds confidence in the model for screening new compounds like nanoparticles.
And takeaway number two relates to the polypyrrole nanoparticles themselves.
Yes. Based on the C. elegans study, under these conditions, the Ppy NPs seemed pretty biocompatible. No major toxicity alarms went off regarding survival, growth, or reproduction.
But they weren't inert. They did have an effect.
Correct. They clearly influenced the pharyngeal pumping rate. And the fact that the effect was most pronounced in that calcium channel mutant strongly suggests they might impact heart rhythm, potentially by modulating calcium signaling pathways.
It's fascinating.
Yeah.
But we need to keep it in perspective, right? This is still a worm pharynx, not a human heart. What about limitations?
Absolutely crucial point. The researchers themselves are clear about this. While C. elegans is a fantastic initial screening tool and great for probing basic mechanisms, it's not a miniature human heart.
Lacks the four chambers, the complex vasculature.
All of that. So any promising findings like these absolutely need further rigorous testing in larger animal models with more complex cardiovascular systems before anyone can even think about human trials. This is an early step.
An important early step, though. It really is amazing how much we can learn from seemingly simple organisms.
It truly is.
So for you listening, here's something to ponder.
Yeah.
How could really leveraging these simple model systems, like C. elegans, genuinely change the game? Could it help us develop safer, more effective drugs, maybe for conditions like arrhythmias, much faster?
And beyond that, what other secrets are hidden inside these tiny creatures? What other complex human diseases might we get clues about by studying them?
Makes you think, doesn't it?
Yeah.
Maybe also worth looking into those 3R principles: replace, reduce, refine, and how they're pushing science towards more ethical and potentially more efficient ways of discovery. Lots to consider.